thank you very much and welcome to the Life Science Technology Day.
Well, thank you very much. What I'd like to do is give you my vision of how there are emerging transformational technologies in biology that are utterly going to transform healthcare, and I think do so in a very short period of time. So when I went to Caltech in 1970 as a newly minted assistant professor, I was really impressed with the complexity both of biology and the study of disease at that time. And that really started me thinking down an evolutionary path that ultimately led to systems biology on the one hand. But on the other hand, it led me to realize that we had to develop technologies, systems-driven strategies that could enable deciphering complexity. And I had the good fortune over a period of much of my career to be involved in what I've called 7 different paradigm changes. And one you've heard about, that was we developed at Caltech 4 instruments and later on 2 additional instruments that could basically read and write DNA. And these things did made 2 important advances. One, they brought to biology for the first time the ability to do high throughput generation of biological information. And number 2, that ushered in ultimately the era of big data and its analytics that we're all familiar with. Now, one of these instruments, the automated DNA sequencer, made it possible to eventually sequence the human genome. And I got invited to the first meeting in 1985 ever held on that with 11 others to kind of pass judgment on whether this was a good idea. And we came to the conclusion, one, that it was technically feasible, although at that time very difficult to think about sequencing the genome, human genome. And number 2, we were split 6-6 on whether it was a good idea, and those against were fanatic. And indeed, going out into the community in 85 and 6 and 7, there was enormous opposition. Probably 80% of the biologists utterly opposed the Genome Project in general because it was big science and seen as destructive of beloved small science. And of course, the Genome Project ironically opened up biology in ways that enabled small science in many different arenas. So what was really interesting, though, is the most opposed institutional organization was NIH, and they opposed it bitterly right up until the very end when it turned out that it was going to happen, and they switched over and became an advocate, and of course played a major role in it and everything. But the Genome Project, for the first time, gave us a complete parts list of all genes and by inference all proteins, and that enabled us to start thinking at a human level about systems biology. And this emergence of systems biology really started in the early 90s and so forth. Now, one other thing the development of the automated sequencer did was it really required bringing together computer science, chemistry, engineering, and molecular biology. And it made me realize that biology really needed to live, that is, leading edge biology really needed to live in a cross-disciplinary environment where the biology could drive relevant technologies and they in turn push the development of the analytic tools for analyzing the information. And I went to the president of Caltech in the late 80s and said, look, I'd like to start an applied biology department at Caltech. And everybody thought that was a great idea, but for the biologists who really vetoed it. So Bill Gates made it possible to go to the University of Washington and start in 1992 the first such department, and it was spectacularly successful. But what was interesting is I'd wanted to build on top of that cross-disciplinary department by that time, a systems biology center. But I found the bureaucracy of a state university utterly intractable for really new ideas. And in fact, one of my most fundamental beliefs is any really novel new idea can almost never emerge from an old bureaucracy because of the constraints they impose. So in 2000, I started the Institute for Systems Biology, and that led to our looking at systems approaches to disease, and that in turn led to these concepts of systems medicine and P4 medicine, a healthcare that is predictive, preventive, personalized, and participatory. And emerging from that shortly came the really interesting idea of scientific wellness and the idea that what was essential in medicine was to generate enormous data clouds around each individual that could be analyzed for wellness and propensity to disease. We'll talk about that in some detail. And then in April of this year, the final paradigm change came when Providence, the largest nonprofit healthcare system in the US, approached me and said, would you like to become our scientific arm and you personally, our chief science officer? So for the first time, I saw a path of how we really could bring P4 medicine to the bedside, and we'll show you exactly what is meant by that. So let me say a word or two about systems biology, just to give you its essence. It's 16 years old now. We have 9 faculty, almost 200 individuals, in a reasonably substantial budget. But what the Institute really is all about is driving the application of systems thinking toward both biology and toward disease. We've developed a lot of technologies, and I'll be talking about some of those. We've played a major role in developing computational tools, many like Cytoscape, very widely used. We have created a cross-disciplinary environment. And I think what is really critical about our cross-disciplinary environment is we really mandate that individuals that come into it learn the languages of the other disciplines so they can work together effectively in teams. And I think we've mastered that very well. We're aware of the fact that most of the really complex problems in biology, and especially in medicine, require the integration of many different skills and technologies and the so-called big science, which is cross-disciplinary, systems-driven, integrative, and milestone-driven, is really the essence of how we operate these days. And a key part of that is leadership. That is a clear vision of the goal to which you're really trying to drive. We're also all about thinking of strategic partners and bringing them in from the outside. I mean, in the world today, you don't need to be bound geographically. If you've got terrific people that can contribute to what you want to do, they can do so from virtually any place. And we're also about transferring knowledge to society. So in the 16 years of our existence, we've started 8 biotech companies. They're all in good shape. Some of them are going to be very important biotech companies, but the idea is that you can use companies to enable the development of a vision more effectively than just doing it in the academic sector, I think is really an important concept. The question you can ask is, what are the major contributions that systems thinking has brought to systems medicine and P4 medicine? And I would say there are 2 major ones. So one is this idea of a dense, dynamic, personalized data cloud, many, many different types of data that surround an individual, and we'll define a subset later that we've actually explored. And the concept is that you can integrate and analyze this data in such a manner for each individual to lead to the actionable possibilities that either optimize wellness and or let them avoid or ameliorate disease. And we'll talk more about that later. Now, we started doing this, as you'll soon see, in 2014. And of course, this is precisely what I think precision medicine should be. And almost in no place is it. Generally, precision medicine is, let's take 1 million people, let's get genome sequence and clinical records and maybe a little bit of personal apps that do quantized self-analyses. But my view of what precision medicine should be is this much more global and holistic approach toward the complexity of an individual human being. From these kind of data, we can then generate the, in a sense, network of networks. Networks operate at many different levels in living organisms, and they're seamlessly integrated in the organism, and they're really the conduits of information for development, for physiologic responses, for aging. And if disease perturbed, they cause disease. And if one can distinguish in key networks the difference between disease perturbed and the normal network, you can get deep insights into the pathophysiology of disease, and you can take completely new strategies to getting biomarkers for detection and drug target candidates for reversal of disease. And we'll talk more about that in just a few moments. So this all went along very nicely until about 2007. We defined systems medicine, and we're beginning to define P4 medicine. But it was clear, again, that there were a whole series of new technologies that needed to emerge. And we found NIH almost impossible to use the support for these because they demanded things were essentially done before they could ever be funded. So I had the good fortune to meet the Minister of Finance for the state of Luxembourg in 2007. And he was, the dominant politician in Luxembourg at that time, and he decided to carry out a transform in the country of its financial bases from a 90% dependence on financial services to bring in healthcare and biotech. And he asked us to write a proposal to help them do that. And we did many things for Luxembourg and the University of Luxembourg, and I won't talk about those here, but what they did for us was to give us $100 million over a period of 5 years to invent the technologies and strategies of systems medicine, of P4 medicine. And these enabled us to invent 8 or 9 new kinds of approaches. And then in 2013, we were really at an interesting tipping point. And, you know, Malcolm Gladwell, the tipping point is that magic moment when an idea, a trend, or a social behavior tips and spreads like wildfire. And we thought we were exactly in this same kind of position. And of course, what really catalyzed it was the Luxembourg Strategic Partnership that enabled these technologies. So here are some of the technologies that we've participated in, and I'm only going to talk about a couple of these. I'll discuss one of them a little bit later on. But my view is third-generation DNA sequencing, single molecule sequencing, is going to, within an 8 to 10 year period, bring the cost of the human genome sequence down to $100 or less. And you'll be able to get a human genome sequence in 10 or 15 minutes. So it's going to be transformational. It'll make your genome sequence a typical part of a patient's record. I would say the second thing, and unfortunately I don't have time to talk about this in detail, is I think single cell analysis is going to allow us to use many new concepts from physics in biology and open up whole new avenues for exploring complexity and understanding it in ways we've never been able to do that before. And we're beginning to see the barest emergence of such kinds of things already. Now, a systems-driven strategy is one where you take one or more technologies, you put it on an adequate computational platform, and you make it basically high throughput. So, and these are all a series of systems-driven strategies, and I'm going to be talking about many of them just to give you an idea of what these things are truly about. So the first one I'll talk about is using mice as a model to understand human disease from their initiation to their termination. And we did this for neurodegeneration by injecting into the brains of mice activated prion particles to cause a neurodegeneration process that proceeded over 22 weeks and to death at that time. And what we did was to analyze the transcriptomes of these infected mice across that time period. And from that, we were able to derive major biological networks that were disease-perturbed and specified the nature of this disease. But I'll just make one really interesting point. When we did the initial comparative analyses, and that was after subtracting away the transcriptomes from normal mice at these different time points, we ended up seeing that a third of the genes in the mouse had changed in this process. And it turns out that only 300 were really involved, 300 as opposed to 7,000 were involved in the neurodegeneration process, and virtually all the rest were noise that arose from irrelevant biological processes. And we figured out how to subtract all of that away. But the important point was they revealed networks that changed dynamically across the period of time. And in fact, we defined 10 networks, and collectively their behaviors explained almost every aspect of the pathophysiology of disease. But one really important point was the fact that the networks became disease-perturbed in a sequential manner. And here I show 4 of the major networks. And the importance of that is the initiation of the first disease-perturbed network occurs about 12 weeks before any clinical signs show up whatsoever. And what you'd like to be able to do is detect this in the blood, and we can. I'll show you how we're going to do it in just a minute. And then be able to reverse that so that it never gets to the point of manifesting itself as a disease phenotype. And I'm going to talk later how we can do this with humans. We have exactly the same kind of options with human beings at this point in time. And the key to getting the biomarkers that let us identify the sequential process of this disease-perturbed network activation, or disease perturbation, was what we called brain-specific blood proteins. And I won't go through the details, except I'll say we have, for humans, about 100 proteins in the blood we can measure with mass spectrometry. And for mice, we have about 90 proteins we can measure in the blood. And what's nice about these things is those 100 human proteins map back into many of the central networks that we know operate in the brain. And a normal individual will have these 100 proteins set at one level. If one or a few networks become disease-perturbed, the cognate proteins from those networks alter their concentration levels in a very specific way, so you can distinguish health from disease, and even which disease you have among those. So we were able to take 11 proteins that mapped into the 4 major networks that I showed you previously. And we were able to show in the blood, delayed only by about a week, the emergence of successive disease-perturbed networks. So that means we can, from the blood, define the nature of what's going on with disease and in various organ systems. We now have organ-specific proteins for about 20 different organs in the human, and it's a major approach to studying biomarkers and everything. So a second area that we'll talk about is another approach to creating biomarkers, and that's an approach that takes advantage of an Institute of Medicine report that came out in 2012 that said, look, there have been hundreds of biomarker papers in Nature, Science, and Cell, and at that time, not a one of them had become a clinically relevant marker. And they said there are 2 reasons for that. One is the genetic polymorphisms of the human population. So when you do biomarker discovery, you should do it in multiple distinct populations so you can subtract out that contribution via polymorphisms. But the second point was that everybody looked at bloods from normal individuals and compared them with bloods from diseased individuals and saw enormous numbers of differences. And 99.5% of those differences were noise. And the question is, how do you filter out the noise from the signal? And essentially, the only way you can really do it effectively is using systems approaches to noise. So what we did with the company Integrated Diagnostics then was to take on the following problem. In the US, in lungs, there are 3 million nodules that are observed a year. 600,000 of these go into surgical procedures. More than half of those turn out to be benign. So you waste money, you waste the morbidity that comes from these surgical procedures. So what we set out to do was get a biomarker that could distinguish the normal lung nodule from the neoplastic lung nodule. And the approach that we took was to, by various systems-driven methods, identify 371 proteins that, for a variety of reasons, we thought should be expressed in the blood. When we made the assays and looked at all of those proteins, we found we could only detect reliably about 190 of them. So we took these 190 proteins then, and we analyzed 72 bloods from individuals with benign nodules and 72 with neoplastic nodules. And what we did was rank order the 109 biomarkers with regard to their ability to make the 144 correct calls. And we found that there were 32 of these proteins that stood out way above the rest in effectiveness. And then we did yet another systems filtering, which was to take these 32 proteins and ask the computer to make 1 million panels of 10, and then scored those 1 million panels against the 144 calls. And then we asked which of the proteins were most cooperative, in the sense they were found most frequently in the panels that had the highest effective scores. And when we finished that, we found that there were 13 proteins that were very effective, that were cooperative. And we went on to do validation for those 13, and we were able to show those 13 proteins had the capacity to identify more than 36% of the benign nodules. And in fact, we went even one step further. That was commercialized, and I'll tell you why it was easily commercialized in just a moment. But we've gone on from the 13 now to develop a 2-panel marker using some new strategies for biomarkers that are actually even more effective. They can do more than 50% of the calls and everything. So what we have then is, one, we've started with about 400 proteins, and we're now down to 2, and we can do the calls that allow us to eliminate more than 40% of the surgeries. And the importance of that to the healthcare system is you save about $4.5 billion a year by avoiding the unnecessary surgeries. And what is interesting is both the 13 and the 2 biomarkers we've talked about here map beautifully into major disease-perturbed networks in small cell lung cancer. So let me just say, I think we really have solved how to make biomarkers effectively. So we've done it for lung cancer. We've, in collaboration with a company in Salt Lake City, done it for preterm birth. Again, a 2-panel that is marker that is very effective at 19 weeks in making the calls of which women will go on to preterm birth. And you can delay the pregnancy with hormones if you know who's going to likely to go on to preterm birth. So it's made an enormous difference. We've actually got a couple of proteins and some microRNAs for post-traumatic stress disorder, soldiers from Afghanistan, where we can distinguish beautifully the two from one another. And this opens up the idea that you can create blood biomarkers for neuropsychological diseases as well as the more physical diseases and everything. And I'll argue later we can make wellness panels to distinguish how healthy you are. And then finally, glioblastoma, major type of brain tumor. We have 4 terrific markers that are going to essentially let us look at very early stages of the disease and follow the progression all the way through and everything. A third technology is one that Jim Heath at Caltech has really pioneered on peptide-based protein capture agents. And these are absolutely going to replace monoclonal antibodies. And together with Jim, we started a company called Indy Molecular that is pioneering these strategies. And the idea is the following. Jim can make a library of 10 to the 6 circular D-amino acid peptides, and he can use a protein to capture low-affinity monomeric reagents that bind to it. And you can use a chemistry called click chemistry to join in appropriate 3-dimensional orientation 2 such dimers to 2 such monomers to create a dimer reagent. And the typical dimer reagent has mid-picomole sensitivity, which is as good as the best monoclonal antibodies, and we can actually increase the sensitivity by adding a 3rd or a 4th monomer if we choose to do so. What Jim has also shown more recently, and this is really a key point, is you can select peptide epitopes as targets for these peptide protein capture agents. And when you target them to a peptide, you enormously reduce the cross-reactivities that utterly plague monoclonal antibodies and most of the assays and complex systems that you talk about. And the other thing that we've done more recently is demonstrated in a series of collaborations with Merck that these reagents are going to be terrific drugs. And we can use them either to block protein-protein interactions or to paralyze allosteric transformations, to do a variety of different kinds of things. But I think these reagents have really terrific features. They're stable. You can put them in an envelope, send them to Africa, and the affinities won't change. You can make them as sensitive as you want and with regard to sensitivity and specificity. They're digital in that if you know the monomers and the nature of the coupling reagent, you can put them all in vitro with copper catalysis and produce large quantities of the material. You can minimize cross-reactivity, and we're now moving up to do very large-scale production. So with these, we can do in vivo and in vitro diagnosis, and we can approach things toward therapy. And I'm quite confident these are going to replace monoclonal antibodies that have all the limitations that you know so well. And the final technology I'll talk about is with a collaborator at Montreal, Mike Tyers, who has really a striking new way of creating very large drug libraries by synthesizing natural products in a really clever way. And the basic idea is he can easily synthesize 1 billion different drugs, and then you can use high-throughput screening to identify which drugs are effective for what kind of uses. And the approach that he's taken is, number one, DNA synthesis is really inexpensive now. And number two, there are an enormous number of natural enzymes out there from all the microbes that have been sequenced in the world. And he has actually synthesized now 1,600 of these synthetic enzymes. And what he's been able to do is put them in blocks of sequences, and then he can put together 4 or 5 or 6 of these enzymes in a combinatorial fashion to create yeasts that have the ability to synthesize a whole series of different natural products. And the cost for doing this is fractions of a penny as compared with drug companies that usually cost $1 to $2 per compound screened and so forth. And in the drug companies, the number of compounds they screen is limited, and often they're in a three-dimensional space area that isn't very relevant as drugs. The natural synthetic products are the really relevant ones. And this is one of my favorite Freeman Dyson quotations. New directions in science are launched by new tools more often than by new concepts. So the idea is that a concept-driven kind of revolution takes old things and rediscusses them in new ways, where a tool-driven evolution discovers new things that have yet to be explained. So these are the technologies we now have at hand to be able to do systems and P4 medicine. So in 2013, 14, there was this convergence of systems medicine, of digital measurements that everyone is doing these days, of big data and its analytics, and of social networks. And that allowed us to define much more sharply exactly what P4 medicine is and how it's contrasted with contemporary medicine. So one, P4 is proactive. Number two, it is focused on individuals and not on populations. Number three, it thinks about wellness as well as disease and not just disease. And number four, it's about creating these dense, dynamic, personalized data clouds, because it is only through those that we can assess both environmental and genetic contributions to an individual's health. And five, we think the old-fashioned way of drug discovery, putting 20,000 patients in a pool and giving them drug or placebo and extracting the information is fatally flawed, because each individual is unique genetically, unique environmentally, and you have no business averaging such different individuals, and you enhance the noise enormously. So what P4 suggests is take 20,000 individuals, each with their unique data clouds, and stratify them based on their own features according to properties you're interested in. Do they respond to a drug or not respond to a drug, or whatever you'd like to do? And of course, this raises the possibility that you can do N of 1 experiments on a single individual that are enormously meaningful in deconvoluting certain aspects of complexity in human disease. And then finally, the social networks are great for education of patients and, frankly, physicians, for crowdsourcing, for advocacy, and all of these different kinds of things. And this slide is really a striking one. It's the top 10 selling drugs in the US today. The orange people are those that respond to the drug. The best response is 1 in 4. The worst is 1 in 25. So you give drugs to so many people that have no effect on them whatsoever. And what we would argue is the way you currently do the drug analyses, you have no clue as how to distinguish the orange from the non-responders. And if we did it the P4 way, you'd immediately have the information to sort out and stratify those individuals that would be responsive versus those that would be non-responsive. And of course, this leads us to this view that healthcare is really about two things. One is wellness, and the other is disease. And I'd argue that today's resources are 98% on the level of disease, almost none to wellness. And my prediction is that there will be a whole new sector of healthcare called scientific wellness, whose market cap in a 10 to 25-year period, I think, will come to exceed that of the old-fashioned health industry or the so-called disease industry. And a point I really want to make is the major applications of dense dynamic personalized data clouds. So one, I'll show you in just a moment, it can be used to optimize health. Number two, if you follow a large number of individuals for a long period of time, you'll begin to see transitions from wellness to disease for all the major diseases. We'll be able to delineate with biomarkers their earliest transitions, and we'll use the networks to identify drugs that can reverse at the earliest stage. I think the preventive medicine of the 21st century is going to be this early reversal of disease. You obviously can use these clouds to study disease, its initiation, its progression, its response to therapy, its return to health or not. And of course, these N-of-1 experiments that I talked about, I think are really going to be key in many areas. For example, I think nutrition is in the dark days. And I think with N-of-1 experiments, we'll be able to convolute many of the complexities of nutrition. So in 2014, I persuaded 108 of my friends to go through a year-long study using these dense dynamic data clouds to study scientific wellness. And this included genome analysis. It included drawing bloods every 3 months to get clinical chemistries and metabolites and proteins. It involved sampling the feces to get gut microbiome ratios and quantized self-measurements with a Fitbit and things like that. And we had actually 3 blood draws across that period of time. We analyzed the data. We got to then actionable possibilities that, again, either optimize wellness and or minimize disease. And a really critical part of this program was the use of coaches to bring the actionable possibilities to each individual. One, to explain what the actionable possibility was, and two, in the context of what their true health objectives were, which the coaches learned initially, to place the actionable possibility in that context. We got 70% compliance because of the coaches, and the coaches made it possible to have people re-sign up for second and third years. So coaches aren't replaceable by apps or anything. The data was really spectacular, and I really liken it to the Hubble telescope, which for the first time could look at the heavens with a resolution heretofore inaccessible. So too with these dense dynamic personalized data clouds, we've looked at normal human biology and disease in ways that I think are really striking and give us deep insights into many things that are going on. For example, one of the things we can do is ask about statistical correlations in 2 contexts. One, at a given point in time for a set of individuals, or 2, across time for the set of individuals. And they actually come to very different kinds of conclusions in interesting ways, which we won't have time to talk about. But the first analysis that we did was to look at the 6 different data types that we had and to ask for each bit within a given type is it statistically, in a positive or negative sense, related to any other data bits in any of the other 5 types? We identified of the order of 4,000 or so correlations. These showed us that systems were interrelated we had no idea about. They made really interesting suggestions about possible biomarkers, possible drug targets, and things like this. They opened up the possibility of doing disease stratification in really striking ways and everything. And in fact, one of the things that we did was to take the hairball network of these 4,000 correlations and then to use a program that successively deleted the edges that represented the lowest likelihood of an association. And what that generated very quickly were clusters of interrelated proteins that turned out to correlate beautifully with physiologic systems. And in fact, we could look at some of these clusters and get proteins that were interrelated to many other aspects of the system that could turn out to be incredible diagnostic reagents. And in fact, we found about 7 or 8 therapeutic targets. And when we really scanned the literature, almost all of them were being looked at in various drug companies that had not yet been announced. But we had probably 20 other possible kind of candidates. So this is an enormous gold mine. The second thing we can do is by using genome-wide association data, we can actually, for each individual, sum up the variants to give them a relative risk for a given disease, in this case, high cholesterol, LDL cholesterol, the bad cholesterol. And what we were able to do then is to take the 108, to bin them into these 5 categories according to their risk. And the first thing we did was to measure cholesterol, LDL cholesterol for each, and show that the disease phenotype correlated beautifully with the disease genetic prediction. We did that for 4 other diseases, so these correlations, I think, are really quite good. So here are a list of the GWAS diseases, many neurodegenerative, many cancer, many different types of things. And you can think in completely different ways about doing clinical trials with groups whose genetic risk you actually know now. The genetics also tell us how to diet, how to exercise, how to avoid athletic injuries. There are more than 330 variants that correlate with various athletic injuries. They tell us a lot about nutrition and inflammation and even heavy metal toxicity. We had 3 of the 108 pioneers with excessively high arsenic levels, and it turned out 2 ate excessive amounts of tuna sushi, and when they substituted salmon for tuna, their mercury levels came right down. And the third had old amalgam fillings in it, and when they were replaced, his mercury levels came right down at that time. And the final area is that of state transitions. And of course, they can be transitions from wellness to greater wellness. And we're using the parameters that change in this index to be able to get a metric for wellness. And we think we can do it physiologically as well as psychologically. We're able to see now some wellness to early disease transitions, and they give us, again, the possibility of reversal that will lead to the preventive medicine of the future. And we're going to show you examples of where we're going to be studying disease in really effective ways with these dense, dynamic, personalized data clouds. And of course, for the 108 individuals, when they agreed to act upon an actionable possibility, the best reinforcement they could see was corresponding predictable change in their chemistries that said, yes, they were indeed improving their health in interesting ways. Here's actually the first clinical tests we did on the 91 individuals to show striking results. 91% had nutritional abnormalities. A lot of these were genetic variants that easily could be corrected with supplements. Many had inflammation, And most of that had to do with really terrible diets. And we saw people with cardiovascular and diabetic risk. Half the pioneers had prediabetes. By the end of the experiment, 7 had moved to normal, and most of the rest had moved over very close to normal. So it really did make a difference. And here's a beautiful example of what data integration does for you. 91 of the individuals had low vitamin D, so we immediately said, take 100 international units. And for a few of the people, they started to move up to normal, but for most, it didn't budge them at all. Then we found that there were 6 variants and 3 genes, each of which blocked vitamin D uptake. If you had multiple of those variances, you needed megadoses of vitamin D to get up to normal. So by putting those 2 parameters together, you can make the right call on how to have people have normal vitamin D levels. And this is just 5% of the individuals in the study had chronic diseases and didn't know it. And one of the diseases was hemochromatosis. And this is a gruesome disease that, if left unattended, costs society an enormous amount of money. And we had an individual come in with arthritis in the ankles that prevented him from hiking. He'd had concierge physicians on the East Coast and West Coast. They could never make the call. The first thing we saw is his transferrin level was high, which meant he had very high levels of iron in the blood. And the second thing we could do was look at the gene that encoded hemochromatosis and show he was homozygous for the variant that causes this disease. And the actionable possibility was simple. You bled him for a unit a month for just a couple of months, and his iron levels came right down. So, and it reversed the symptoms of arthritis and all of those things. So the correlations, the genetic determination of risk, and the state transitions all are effective both in helping individuals with regard to their disease, but they're really going to be transformational in changing how the healthcare industry, pharma, biotech, nutrition companies, diagnostic companies carry out things, because what the dense dynamic data clouds do is they allow us to deal with noise in a way we've never been able to deal with noise before. So what about the experiences of the 108 pioneers? 50% of them went in skeptical this would do anything for them at all. Almost all came out convinced it was actually a unique lifetime experience. And many of them went on to Aerovale, a company I'll talk about in just a moment. But learning you could control your health was revelatory. Learning that genetics didn't determine your destination, just your potential, and you could avoid many genetic limitations in reasonable ways. And of course, the really interesting one is they all came to understand that their wellness was only a relative thing, and these actionable possibilities actually could really change things. And it's kind of like a staircase here, where the average person is rather low on the wellness arena, and it's these actionable possibilities that will gradually move you up these steps. And of course, the real aspiration is to get up here and mimic the elderly. These are 1,000 individuals Eric Topol at Scripps has studied that are 90 or older, have never been in the hospital a day in their life, are perfectly active functionally, mentally. And so if we can aspire through scientific wellness to elevate people to that level so that you can go into your 90s effectively, what this means then is that scientific wellness, I think, is going to be a lifetime journey. What's been interesting is, either from the literature or from different types of data integration, we've added enormously to the number of actionable possibilities over the past 3 years. And that will continue to increase and give us the potential for moving things up this ladder. So we started a year ago a company called Aerovale that was directed at bringing scientific wellness to the consumer. We believe with this company will have of the order of 10,000 individuals on this dense dynamic data cloud regimen within the next year to 18 months, and really begin to see the transitions we've talked about before. And the data we're analyzing in many different ways. I've given you just a small taste of what we've done. And with ISB, we're really pushing, bringing the cost of the assays down to a level that will be much more reasonable than it is now, of the order of $4,000 to $5,000 per individual per year. And we're also interested in developing analytics that are going to integrate these very diverse types of data, and creating the metrics for wellness that we think will be really useful in the future. As I said earlier, in April of this year, we merged with Providence, which is a very large healthcare system, the largest nonprofit healthcare system. And ISB became their research arm. I became chief science officer of this endeavor. And what this did was open us up to the possibility of bringing these systems-driven ideas for translational medicine right to the bedside. And I'll talk about 5 different translational pillars in just a moment. Providence is an enormous system. It's in 7 states, 107,000 employees, a $20 billion operation. It has 3 million electronic medical records, which are wonderful for beginning to extract really exciting information from it. As I said, it's the largest nonprofit healthcare center in the U.S. So the 5 translational pillars. 1, I just want to emphasize that the partnership between ISB, Providence, Aravale, and a company that has the ability to extract from Epic, the classic clinical database in the U.S., anonymize patient records and put them in the cloud in a manner that they're queryable by Jupyter workstations, gives us enormous opportunity for the future. Aravale is going to do all the dense dynamic data clouds and the coaching and help with the analytics and so forth. So I just want to remind you that we have enormous power in going after these diseases. I've talked about understanding the dynamics of disease-perturbed networks and what they can do. Blood has become a window, so we can readily get biomarkers to look at transition states. We have this power of peptide protein capture agents to create ELISA assays that are highly miniaturized, and we'll be able to do literally thousands of measurements on a droplet of blood in the future with appropriate microfluidics and nanotechnology. We can use these dynamic disease-perturbed networks to make computational predictions, both about biomarkers and appropriate drug targets and so forth. And the new approach we talked with Mike Tyers of drug discovery is really exciting, and we've talked in detail about the dense dynamic personal data cloud. So these offer a palette of enormously powerful tools then to attack these 5 pillars. And I'm just going to give you a very quick summary. For scientific wellness, what we're doing is there'll be 2,000 employees from Providence that will get the Aravale scientific wellness for 3 to 4 years, and 2,000 employees that get whatever wellness they gotten before from Providence. And we'll follow them across this period of time, both to ascertain how their health has been improved in the scientific wellness group, and we'll be able to look at the economic savings scientific wellness provides over just the regular kind of treatment. And that's really key to bringing this opportunity to the payers and persuading them actually to pay for it, rather than have, as we do now, the individuals pay for it. A second area is Alzheimer's disease. And again, we want to do 2 things. One is readily be able to identify early transition states of Alzheimer's disease and reverse it using a regimen that Dale Breslin at UCLA and the Buck Institute has designed from a systems point of view about how to facilitate neural synaptic interactions and so forth. And this 36-point regimen has supplements and vitamins and drugs and exercise and a whole variety of things. With 124 individuals, he's had remarkable success in that reversal. And we just want to put this into a regular clinical trial and show we can reverse the early stage of Alzheimer's. And then with 200 individuals at highest risk for Alzheimer's, we're going to follow them with the dense dynamic data clouds. The second they begin transitioning in any way toward the initial stages of Alzheimer's, we'll start treatment with the Breslin protocol to show we can prevent high-risk people from ever moving into that realm of Alzheimer's disease. Type 2 diabetes, we're putting together 200 individuals that are diabetic in one arm of a study, 200 that are pre-diabetic, and then 200 controls. And we're going to follow them again for a 3-year period with a whole series of supplement additions and opportunities to change behavior. We're going to take 200 individuals that have been newly diagnosed with breast cancer, put them on the personal data clouds, and then follow them through therapy, and immediately after therapy, try and bring them back to wellness as rapidly as we can in the context of what we understand about scientific wellness, but especially focusing on about 10 complications that come routinely through women that have been through post-breast cancer treatment and so forth. And then finally, we're going to use glioblastoma as a model system. I've always felt cancer should really be treated exactly like AIDS is. That is, you should start on day one with triple drug therapy. And with glioblastomas, we can easily capture tumor stem cells from resected tumors for each individual, amplify them, put them in 384 wells, and then analyze a whole series of 20 cancer drugs for each individual, which works best, and then take the top 6 or so and put them together in a combinatorial manner and see which of those works best. And we'll try this for a year or two and see what the combinatorial strategy actually does. And we'll be doing many other things, but we don't have time to talk about that. So I think we really are bringing P4 medicine to the healthcare system. I think we really are going to use these dense dynamic personal data clouds as an assessment of the integration of genetics and environment for the individual, be it in well people and or in sick people. I think we really can optimize health. I think we can identify the earliest transitions and begin to use all the strategies we've talked about to reverse them. I think scientific wellness, as I've said, is really going to be a lifetime journey. And my guess is that the health savings from this are absolutely going to be enormous. And I think we'll have that data in 3 or 4 years. We are going to create a whole new scientific, a sector of scientific wellness. And I think it will quickly become populated by many different companies. I think the data are going to transform how the healthcare industry absolutely operates. And I think finally, if we can really reduce the assays and optimize the analytics, we can begin to think about bringing scientific wellness to developing as well as developed countries, and think in the future, perhaps a 10-year time span of a democratization of healthcare that really was inconceivable even a few years ago. So, thank you very much.
Thank you, Lee, for an exciting and view into the future. Now, Lee will be leaving rather soon for the airport, actually. So if there's any questions, we have a short slot of time to ask some questions. And now is the opportunity.
Okay, thanks. Excellent talk. You mentioned Alzheimer's disease. Have you already discovered blood biomarkers for that based on this brain-specific blood idea, or is that the plan?
That is, we're adding the brain-specific biomarkers to the data gathering we'll be doing at this point in time. So we haven't done that yet, but that will be done in the next year.
So I'm a bit skeptical that you will go do large savings. So why, even if you are longer in life, how do you know that the time you think is shortened? So basically, you should be. Is there
Well,
evidence for that?
I think the biggest savings are going to become from people that transition into chronic diseases. Hemochromatosis costs society an enormous amount of money. We've actually, in the 108, reversed this pathway for 3 different individuals. And you can make estimates, but they're over the lifetime of the individual, you're talking about many, many hundreds of thousands of dollars. And I think you can do that with a lot of chronic diseases. I think you can do it with reversing diseases at the earliest stage before they, if you add up all the different kinds of things that you can do, I think they soon come to be large numbers. But we'll see what happens after 3 or 4 years on the scientific wellness, and we'll see if you're right. I think almost certainly you're going to be wrong, but we'll have the data to show it.
Thank you very much for a very inspiring talk. I'm here. Sorry.
Let's see. Oh,
Lee,
okay.
I'm here.
Okay, okay.
I have a question regarding your wellness study and the data you're generating. There's a long way to go from a correlation network or omics data system to giving data back to individuals. So what kind of data are you giving back, and how do you explain those types of data to those kind of normal, non-scientific individuals?
So the agreement we had with 100 pioneers was they could get back any part of the data that they wished. What we gave them was the data that was relevant to actionable possibilities, and we put out dashboards that showed where they were and where they were going across time and so forth. But as you know, when we moved to Airvale, there was enormous constraint imposed by the FDA because a commercial wellness company cannot do disease-like activities. 23andMe got shut down for a while for doing that kind of thing. So one of the enormous benefits we have with Providence is we'll be setting up clinics to bring the pioneers in Airvale all the disease-related information that we really have. And it'll be up to them. People can come and take advantage of this or not. But in general, if you want the genome sequence, we can give it to you in whatever form you think is. We've not had anybody actually ask for a whole genome sequence. I'm not sure what they do. But in general, we're agreeable to giving the data if that's what they wish.
Okay, I hate to interrupt, but I think we need to
Better go.
get going.
Yeah.
So thank Lee again